Hey, everybody. Today we're going to talk about a really fascinating, important subject, genetically modified organisms, and particularly the new use of CRISPR technologies to create them. Is very, very dangerous. Probably the leading global thought leader on this issue, on the health dangers of genetically modified organisms, GMOs, over the past 27 years is Jeffrey Smith, who has authored 2 bestsellers, directed 5 documentaries, delivered 2,000 lectures and interviews in 45 countries, trained 1,500 speakers, and organized more than 10,000 grassroots advocates. He has counseled world leaders on 6 continents, and his meticulous research presented at medical conferences has inspired thousands to prescribe non-GMO and organic diets. The success of GMO education movement he pioneered is measured in part by the fact that 48% of the world's consumers now acknowledge that GMOs are unhealthy. In recent years, Jeffrey has been sounding the alarm about a new genetic engineering techniques, including gene editing. Unregulated GMO 2.0 can cause health and environmental disasters. The most urgent danger comes from genetically engineered microbes, including the use of CRISPR technology. So thank you, Jeffrey, so much for joining us. And, you and I have spent many, many years in the trenches fighting against Monsanto. But tell us, generally speaking, before we get into GMO 2.0, let's talk about sort of the history of GMOs, because they were promised originally we were going to get a green revolution, and that if we had GMOs, we could increase plant productivity and reduce the amount of chemicals that we use in agriculture. But actually, the GMOs have vastly increased the amount of chemicals that we use in agriculture. And can you explain kind of how that works?
Sure. The original GMOs were created by Monsanto Company. They were selling Roundup, as you know, and it has glyphosate as its chief poison, and that was going off patent in 2000. So they genetically engineered soy and corn, and then later cotton, canola, sugar beets, and alfalfa, putting in a gene from bacteria. Now, the bacteria survived in the presence of glyphosate. Glyphosate is an antibiotic. It kills a lot of bacteria, but this bacteria survived, and they figured, okay, let's put it in the food supply. So they took the gene out of the bacteria and put it into the crops that allowed the crops to also survive what would otherwise be deadly doses of Roundup. So it allowed farmers to use Roundup by spraying over the tops of already maturing soybean fields and corn fields, and it wouldn't kill those crops. So it was a way to weed easier. To spin it, as you said, Monsanto and their PR firm and the other biotech companies claimed it would increase yields, which it hasn't. It would feed the world, which it actually works against. They said it would reduce the use of agricultural chemicals, and it skyrocketed in these herbicide-tolerant crops. And most importantly, they said it was safe. And the FDA said it was safe, but it turns out it wasn't the FDA scientists. It was the political appointee, Monsanto's former attorney, Michael Taylor, who was in charge of GMO policy at the FDA, and he diverged from what the scientists said, claiming falsely that they didn't know of any difference between GMOs and non-GMOs, and therefore they can go on the market without any testing or labeling. And then 9 years later, from lawsuit discovery, we realized it was a fraud, and that the overwhelming consensus among the scientists working at the FDA was exactly the opposite. The GMOs were different and dangerous.
Yeah, and this same thing happened in our lawsuit against Monsanto when we discovered that the head of the pesticide division at EPA, Jess Rowland,
yes,
for a decade was actually secretly working for Monsanto. So they had both agencies that they had nailed down. They had FDA with Michael Taylor.
Michael Taylor. Yeah.
And then they had Jess Rowland at EPA, and in that way, they were able to subvert democracy, subvert the political system, subvert science, and give protection to their industry. As you pointed out, they started using Monsanto, I think, originally in 1973, when their other principal product was DDT. And of course, they were making Agent Orange at that time, too, and spraying it all over Vietnam. But they made DDT EDT got banned in 1973, and they needed a new agricultural product to replace it, a flagship product. Then they found glyphosate, which was a tank descaler. It was used to get corrosion and calcification out of the inside of underground tanks. And they found out that it would kill, or somebody threw some of it out in the yard one day and everything green died. They figured that's a great herbicide. And originally, for the first 30 years, it was applied by men with, they would hire a lot of farm workers with backpack applicators and a little spray gun, and they would walk the corn rows early part of the season. So they weren't spraying it on finished crops. They were spraying it on the weeds that were competing with the corn as the corn first got out of the ground. Once the corn got a foot or two high, the weeds could no longer catch up with it. But those men with backpack sprayers would spray the individual weeds and would kill them, other than having to go with the spade and pull them out of the ground. And then when they discovered Roundup Ready corn, and they put that little gene in it, in the corn seed, so that the corn was now immune to glyphosate, they can now fire all of those workers and hire one guy in an airplane with tanks on it and spray the entire landscapes with glyphosate, and anything green in those landscapes would die except the corn.
And let's talk about that Roundup Ready corn, because it's interesting that the most long-term comprehensive study on any genetically engineered food that we eat for rats was done on Roundup Ready corn. And they found that when they took corn that had been sprayed with Roundup and fed it for 2 years to rats, they developed multiple massive tumors, early death, and organ damage. But this study was designed even in a more advanced way to determine whether it was Roundup that was causing the problem or the genetically engineered corn. So another group of rats were fed the Roundup Ready corn that had never been sprayed with Roundup. And they developed multiple massive tumors, early death, and organ damage, which means maybe it was the GMO. But the third group was fed Roundup in the drinking water and just ate regular corn, and they too had multiple massive tumors, early death, and organ damage, and the control suffered none of these. And this was Dr. Seralini, and as you probably know, he was just lambasted and threatened and discredited by the echo chamber of Monsanto's front groups and pseudoscientists that were paid directly or under the table. And that has been the history of GMOs. In my book, Seeds of Deception, I open with the story of Arpad Pusztai, who discovered that the process of genetic engineering generically, no matter what particular gene you put in, can cause potentially precancerous cell growth in the digestive tract, smaller brains, livers, and testicles, partial atrophy of the liver, and damaged immune system in his rats. And when he went public with his concerns, he was a hero at his prestigious institute for about 2 days. And then a phone call, allegedly from Monsanto to Bill Clinton, and then to the UK Prime Minister's office, and then to his director, resulted in him being fired from his job after 35 years and silenced with threats of a lawsuit. So he became a poster child for what happens if you dare to discover problems with GMOs or even question their safety.
Yeah, and so they have Monsanto and the big chemical producers have control over the political system, and they maintain that control through a variety of mechanisms, including direct contributions to political candidates. But also they have control over other institutions in our society as well, including the media. Will you talk about that a little bit?
Yes, in terms of the media, we can see originally when bovine growth hormone from Monsanto was being pushed, they hired a PR firm to rate all the coverage, and they created friendlies and a list of enemies, and the friendlies were rewarded and the enemies were threatened, and leaked documents showed that they bragged about how they got certain reporters kicked off the case at the New York Times or elsewhere. They had a 4-part news series in a Fox TV affiliate in Florida stopped from a threatening letter from Monsanto's attorney to Fox, promising dire consequences to Rupert Murdoch and his business. And that was just getting started. With GMOs, the mainstream media largely had a love of.
By the way, those Florida reporters were also punished. Their careers were destroyed. There were a pair, two Florida reporters who were
Steve and Jane. Yeah,
Yeah.
they basically what happened was they had a contract and they weren't allowed to be kicked out at that point because they had a contract. So the lawyer for the Fox station, not the local one, the main one, started to bring them through a series of rewrites. And it was not on the basis of what was true. It was on the basis of what they can get away with legally. And over a series of months, they did 81 rewrites. And the moment that the contract gave them the opportunity to fire one of them, they fired him. And they ultimately redid the entire series favorable to bovine growth hormone and ignored all of the controversial evidence that these investigative reporters found. And Jane Aker was unable to get another job as an anchor. I've interviewed her and followed her a bit. So the concept of leaving reporters and scientists in their wake is something that happens not only in the United States, but all over the world. Sometimes I interview the people who are in part of the approval committee for GMOs for their country, and there's usually a majority and a minority. The majority was put there from the influence by industry and often pro-GMO governments. The minority may be representative of civil society or independent scientists. And the ones that talk to me from that side say it's so frustrating because those that approve it do not even look at the data. They just rubber stamp all the approvals. I talked to one of the leading scientists in the world, PM Bhargava, who was put on the approval committee by the Supreme Court of India and said, please evaluate whether this is true or a facade, as is claimed in a citizen's petition. He came back a year later and said, it's totally facade. No GMO in the world has been properly tested. There's about 30 areas that they need to be studied. Only about 10% have been studied, but they've been done by industry themselves in such a poor way, they're meaningless. No GMO should be on the market right now. What's interesting, Robert, is that when you look at the amount of GMOs in corn and soy in the United States on a graph, and it moves up in a particular slope, and the amount of glyphosate-based herbicides sprayed on those, and then you compare it to diseases, there are about 40 different diseases and conditions that have been rising in parallel. Digestive disorders, cancers, ADHD, autism, hypertension. What we now see is a smoking shotgun. Alone, these are just correlations. They're not causations. We can't prove. However, when we look at the animal feeding studies, the animals suffer from precursors to these same diseases. When we look at the modes of action, we could predict these same diseases. When we talk to scientists, or when we talk to veterinarians and talk about the pets or the livestock, they talk about these diseases as becoming prevalent in the animals soon after GMOs and Roundup were introduced, and especially the doctors. I've spoken at about 24 medical conferences, and now I'm hearing from doctors, and have for more than a decade, who've taken a large number of their patients and put them on non-GMO and organic foods, which don't allow either GMOs or Roundup, and there's an astonishing improvement. In fact, we surveyed 3,256 people who got better from 28 different conditions when they switched to non-GMO and largely organic diets.
And what did GMOs do to productivity in terms of agricultural productivity, agricultural yields?
There's actually a yield drag for soybeans because the process of genetic engineering generally causes massive collateral damage and takes energy away from the growing process. They haven't figured out how to increase yield with genetics because it involves lots of different genes, and they've gotten lucky with single genes being put in for single traits, which is very rare. Usually genes work in networks or families. So they haven't been successful at improving the yield in general. If you look at just the corn, there's been about a 2% increase per year because of the impact of the corn on the corn borer. In the corn, they put an insecticide. In soil, there's a bacterium called Bacillus thuringiensis, which produces a toxin which kills caterpillars. It breaks little holes in their guts and kills them. So they take the gene out of the Bacillus thuringiensis, put it into the corn. Now the corn is a registered pesticide. It's an insecticide, and it kills insects, and we eat the corn. Now, the spray is something that washes off and biodegrades in the sun. When it's genetically engineered, the amount of that Bt toxin is thousands of times greater than the spray. But even the spray has shown immune system responses and allergic reactions in humans. When it was sprayed over the Pacific Northwest for gypsy moth infestation, 500 people reported sickness and disease, and some had to go to the hospital. Now, farm workers handling the Bt cotton, the cotton and genetically engineered to produce the Bt toxin in India, are reporting the same symptoms as those that were sprayed with Bt. But it gets worse. We eat that Bt toxin in the corn that we eat. In high concentrations in petri dishes and laboratories, it drills holes in human cells that look exactly like the holes that it kills insects with. In Canada, they found 93% of the pregnant women tested had Bt toxin in their blood, 80% of their unborn fetuses. How did it get in their blood? Possibly through the holes that it drilled. Now, it should wash out of the blood very quickly. So why was 93% of the Canadian women tested containing Bt in their blood? Well, the people who did the study suggest that maybe it was the milk and meat of the animals that they were eating, because those animals ate a lot of Bt corn and Bt cotton as part of their diet. But there's another plausible explanation. In the only human feeding study ever conducted on the commercialized GMOs, this was on soy, they found that part of the gene inserted into the soy, that bacterial gene that allows it to resist death from Roundup, had transferred into the DNA of bacteria living inside the people's digestive tract. Now, they don't know if it was continuing to function. As soon as this was reported, the pro-GMO UK government stopped the funding so they couldn't find the answer. But imagine for a moment that you're eating a corn chip, and the corn chip has genes that produce the Bt toxin. Now imagine if that gene transferred to our gut bacteria and continued to function. It means that our intestinal flora could theoretically become living pesticide factories. And that may be why 93% of the pregnant women in Canada had the Bt toxin in their blood, because it was being produced inside their own digestive tract. This is not hard to detect. No one has done the research. We can't verify that it's happening, but there's certainly indications that the digestion is deeply affected by those that eat GMOs. In fact, 85.2% of the more than 3,000 people that we surveyed reported getting better from digestive disorders when they made the change in their diet.
Yeah, I mean, these are the kinds of studies NIH should be doing, and we should have the answers to all these studies. And, if I manage to get into the White House, it's the first thing that I'm going to do is to, go over to NIH and say, let's get definitive answers on all of this right away. And, so that we can shield Americans from these toxins and shield our children. What about the, if Bt toxins are putting holes in the stomach of caterpillars, and that may account for the disappearance of all our butterfly species. And I don't know if there's anybody's made that connection, but, and how about bees and other pollinators? Do we have any good data on those?
Yes. First of all, in terms of the Bt toxin, one of the Bt varieties was tested years ago and showed that it could kill off the larva of the butterfly, and that was quietly removed from the market. However, one of the big problems with monarch butterflies is that they grow on plants that are killed by the Roundup in the Midwest. So they don't
like milkweed.
Exactly. And so the fact that you have the Bt toxin, which might be causing damage to the insects, and the lack of habitat so the larva can feed, has turned out to be a disaster. Now with bees, we know that Roundup or glyphosate is an antibiotic. And it's an interesting antibiotic in that it kills the beneficial bacteria, but not the nasty stuff. So lactobacillus, bifidobacteria, things that we need, things that we want in our yogurt, etc. Bees need these beneficial bacteria to digest. So when you look at colony collapse disorder with the death of all of these bees, if you think of the neonicotinoid insecticides, which some people blame for it, it wouldn't explain all of the symptoms. One of the symptoms are they seem to starve to death even though there's plenty of food, because they perhaps can't digest it. One research study showed that the bees lose the ability to navigate back to the hive. One study showed that in environmentally relevant levels of glyphosate in the study, there was about a 30% death rate in the hive. And there was a study by a company called Biologics that was looking at the impact between Roundup and these bees, but it was purchased by Monsanto and they stopped doing those studies. But
That company is like, I don't know, it's the dark empire.
I'll tell you, everywhere in the world, because as you read, I've spoken in 45 countries, and the activists usually greet me and give me an orientation and tell me which divisions of the government are now being controlled by Monsanto. And sometimes part of the government brings me in to try and stop the takeover of another part of the government by Monsanto. So they're now bare, but they're still doing the same thing.
Let's talk about, what about in terms of productivity of the yields? I think, what is it now, 85, 95% of the corn produced in the world is now Roundup Ready corn, right? It's a huge amount of it. I
Yeah,
don't know.
about 95%. I know the US number. I don't know the world because there's vast areas in the world that don't allow GMOs. There's
Okay.
very little GM corn growing in the EU. That's in Spain, but about 95% in the United States.
And
And there was a study done in terms of yield and feeding the world that was sponsored by the UN and many other groups. It was called the ISTAD report. It came out in 2008. And they had over 400 scientists working on it, and it was over 2,500 pages. And they evaluated massive numbers of data in over 100 countries. And it's been signed on to by 59 countries since. And it determined that GMOs have nothing to offer to feed the hungry world, to eradicate poverty, to create sustainable agriculture. They described it as a solution looking for a problem or a problem looking for a solution. It just didn't fit. It did not increase yields. It did kill the biodiversity. It was a tool for corporate control. It didn't allow farmers to save their seeds. It damaged the soil bacteria, eliminating regenerative agricultural processes. And so it was a disaster. And that wasn't even looking at the health issues. However, they have been pushing this concept that it was needed to feed the world and that it was going to increase yields. And that takes money away from the more appropriate technologies. And this report determined that agroecology was appropriate because that can double staple crops in developing countries. In one of my books, I described a study with about 12 million farms using agroecology, and it was a 73% increase in yield. And when you think about GMOs as not increasing yield, but being pushed as the solution, it's tremendous hypocrisy, and it's very dangerous to work against the real solutions.
But American farmers seem addicted to it. And why is that? And how do you break that addiction?
Well, you mentioned earlier that there's a lot of control over the government and the media. Farmers are also another area where Monsanto and the biotech industry exert unprecedented control. When you think of what information a farmer gets, there's the land grant universities that provide extension agents that give advice to the farmer. The land grant universities receive money from the biotech industry, which then determines a research agenda. So the extension agents basically say which seeds and which sprays. The farm journals and the farm radio get supported from advertising from Big Ag, and they're not allowed to run anti or even true stories about GMOs. I was told by one reporter, I write for a farm magazine and I'd love to figure out where I can publish an article about your work. I said, can you put it in the farm journal? And she said, no way. They'd never let that happen. There's also a kind of a messaging where GMOs and Roundup are the future, and anyone who's against it is anti-science. There's also a reduction in the availability of the best producing seeds in the non-GMO varieties. There's also a kind of a pride in having the rows between your soybeans completely clean, which only happens when you have the Roundup Ready version. There was also an effort to sue farmers who dared to not plant Monsanto seeds. Then they would sometimes get visited by Monsanto's crop cops who would find contamination, sometimes even in a farm that wasn't even theirs, and then they would get a note saying you must pay us $70,000 for using our seeds illegally or we can sue you. And now there's something called dicamba and dicamba resistant crops, where dicamba can be mixed with the glyphosate, but it can volatilize, rise into the air, and travel, and then land on nearby crops, causing damage. Millions of dollars of damage have occurred. There's been all sorts of lawsuits. It's been up at the EPA. And some farmers realize they have to use the genetically engineered dicamba resistant varieties, or their neighbors who are using it and spraying with dicamba could destroy or damage their crops. So there's a whole nexus from cultural, scientific, economic, etc.
Do you have any idea kind of an exit ramp off of that? If you had a president of the United States or somebody at USDA who wanted to really change that system and change towards regenerative agriculture and help farmers wean off of those chemicals, how would you do it?
Well, I've heard stories, for example, about one GMO corn farmer who worked with a friend of mine, a professor, to implement a regenerative agriculture program. And after the first year, he was making more money and needed less Roundup. And so he said, I can get away with maybe 20% of the Roundup, maybe not at all. And he was doing better. It turns out these agricultural inputs require a level of disability in these crops in order to be on the treadmill for chemicals. If you have healthy soil, it can sequester carbon, it can fix nitrogen, it can produce all sorts of components that are needed for a healthy crop, which can then stave off the plant diseases and improve the yields. But when you have sick plants on denuded soil, you need the fertilizer, you need the pesticides to kill off what would otherwise be naturally killed off. So if research dollars were put into that, demonstration farms, and lots of them in different areas so that the farmers could see other farms in their geography, in their crop, and they can see an improvement per acre in their bottom line. And there was a financial support system to help farmers transition into that, both with the training, the loans, etc., and especially if they want to go to organic for the 3 years during the transition, then it should be pretty easy. They've done some transitions in Germany and other places that were effective, but nothing as big as you and I would probably want.
Okay, tell us about GMO 2.0.
One of the factors of genetic engineering that's important to understand is that the process of inserting a foreign gene or just changing the order or knocking out a particular gene causes massive collateral damage. We know this from the traditional GMOs. There's could be hundreds of changes in proteins and metabolites. There's a new allergen in one of the BT corn plants. The Roundup Ready corn has higher levels of putrescine and cadaverine related to not only the foul odor of rotting dead bodies, but can also link to cancer and allergies. So the collateral damage is dangerous for consumption, and it's unknown what its impacts are in the environment. With gene editing, it also has massive collateral damage, and I'll explain what it is in a minute, but it's important to understand just how dangerous and damaging it is. The journal Nature described it as chromosomal mayhem. So here's what CRISPR is, which is the most popular gene editing technique. Imagine a molecular scissors designed to cut the DNA and a guide telling the scissors where to cut. The guide has a sequence, it looks for the sequence, it matches, the cut is made. Sometimes the cut happens in a dozen other places that are not supposed to be cut. Sometimes additional DNA is added to the cut DNA when the cell repairs it. So now you have mouse DNA or goat DNA or bacterial DNA added in there. Sometimes there's accidental deletions, additions, even something called chromothripsis, a shattering of the chromosome, which rejoins in a haphazard manner. The biotech industry has been pummeled by those of us pointing out the drawbacks and dangers of GMOs. As you said at the beginning, about half the world's population believe correctly that GMOs are not safe to eat. So they've come with a global campaign to convince governments that gene editing is not something that produces GMOs. It's basically a precision breeding method. And they pretend that it is safe and predictable and should not be regulated at all. And they've convinced the US government, the Canadian government, the UK government, Japan, Australia, India, many in South America. And right now, in many of these countries, you can gene edit an organism and put it into the food supply or the environment and tell no one. Now, who's going to do it? Well, CRISPR. CRISPR labs cost less than $2,000 on the low end. That means that you can create a GMO in your basement, especially microbes. But it also means that all of the high schools that are going to get CRISPR labs are going to end up producing these new GMOs. There is a gene rush going on where everyone is looking to find their patent and their solution with this gene editing technology. Now, on the biggest scale, there's going to be an incremental contamination and corruption of nature's gene pool. Ultimately, if we don't curtail it, it's a replacement of nature, the end of biological evolution as we know it. So future generations will not inherit, as we did, the products of the billions of years of evolution, but instead they'll inherit a hybrid natural products and laboratory products from a technology prone to side effects. So that's the bigger picture. As a nonprofit institute, our Institute for Responsible Technology, and that's responsibletechnology.org, we're trying to figure out where we go first. And so we ask the question, what's the most dangerous of organisms to genetically engineer? And it's obvious, it's the microbes. As you know, the microbiome inside our bodies is critical to health. 80% of human diseases are linked to disorders in the microbiome. We outsource about 90% of our chemical and metabolic functions to the microbiome. We can get away with 23,000 genes in our cell, less than earthworms, because we use the genetic information of the 3.5 million genes in the microbes living inside us. There's an elaborate way that infants get delivered a microbiome from the mother and it gets fed from the milk. Part of the milk is inedible by the child. It's designed for the microbiome. If the child gets sick, their salivary microbes change, feeding back through the breast, change the milk, and it helps heal the child, and it reverts back to the other milk when the child is healthy. It is an elaborate, incredible awe that this micro Jedi army works on our behalf. And it works even in a more complicated way in the soil, sequestering carbon, creating a support system for the entire ecosystem. And we've only been able to characterize maybe 1% of the trillion microbes out there. And yet, we do know that when you release a microbe that's genetically engineered, if it survives in the wild, it can travel and mutate. Microbes also can swap genes with other microbes. So now the thing that you created in high school class as an experiment or in some other business or university is now occupying inside the DNA of 10,000 different types of microbes in 100,000 different ecosystems. We don't know what the impact will be, and yet there's virtually no mechanism to prevent that release. No regulatory agencies are in charge of what happens in a high school. Very little research is required for release on the commercial level. And we're of the belief that this is an existential threat that must be handled. And we point to two genetically engineered microbes that were almost released years ago, which could have had potentially catastrophic outcomes. There was a microbe that's popular on the roots of all terrestrial plants called Klebsiella planticola. And it was genetically engineered to turn plant matter into alcohol. It was a well-meaning idea to send it out to the farmers who normally burn their crops after harvest. Instead, rake it up, put it in big barrels with the genetically engineered microbes, and turn it to alcohol. After 2 weeks, you can open the spigot, put it in your tractor, sell it off farm, and then use the nutrient-rich sludge on the bottom to spread on the field as fertilizer. It was a sound idea, except a graduate student got permission to study this genetically engineered microbe, and one day walked into his lab on a Saturday morning, and all of these wheat seeds that he had planted on the soil that had been mixed with the genetically engineered bacteria were dead. It had turned it to alcohol. It was just slime on the surface. The natural controls had no problem. This day was 2 weeks before the company was going to release this outdoors to see how far it would travel. This graduate student's advisor, Professor Elaine Ingham, spoke about this near tragedy at the UN. She was approached by EPA whistleblowers who told her just how far that genetically engineered microbe might have traveled, because the EPA did a study, according to the whistleblowers, that had not been publicly acknowledged, where they released genetically engineered microbes in a field in Louisiana and checked to see how far it spread. 11 miles the first year, another 11 miles. They stopped funding it, but certain employees at the EPA continued to monitor, and eventually they found it everywhere they looked, all over the planet. So I asked Dr. Elaine Ingham, in a film that's available at responsibletechnology.org/takeaction, what would happen if it spread, if it survived, maybe if it displaced its natural Klebsiella planticula counterpart. And she said, quite soberingly, it could theoretically end terrestrial plant life. Now, this is a theoretical example. We don't know if it actually would have been a cataclysm. There's another genetically engineered microbe that was going to reduce the amount of frost on potatoes and strawberries, and it was about to be released and then stopped by court. And it could have theoretically changed weather patterns, because the natural version creates raindrops and snow and sleet. And this genetically engineered version didn't do that. It became impotent. If it had replaced its natural counterpart, it could have changed the balance.
That is terrifying.
Yeah, it is.
I read a brief article today that Bill Gates now has a factory in Colombia that's produce 11 million mosquitoes a week now that are being released. But there's I don't know, hundreds of millions of them in this factory, but they're spreading them all, and they have it's basically a flying vaccine. It's supposed to inoculate people against malaria, but it just seems like, how nutty would you have to be a regulator and approve that?
I know. I remember talking to one of the senior scientists of Oxitec, a company that was trying to get their genetically engineered mosquitoes released in Florida. We were both testifying in 2014 at the Mosquito Control Board. And in the lobby, I said to him, you're going to change the gene pool in ways that you have no idea. He goes, oh, no, it won't survive. It's designed to basically self-eliminate. As soon as they stop releasing them, it'll just disappear. Well, they went to Brazil and they found that in 60% of the samples, 3 years after they were releasing it, there were genes from their genetically engineered version that had mixed with the natural. But I also said to this man, Derek Nemo, I said, have you ever tested the saliva of those mosquitoes in case they bite someone? And he said, we're just now testing to see if the saliva contains that new gene produced inside the mosquitoes. And I'm thinking, it's a little late. You've released it in 4 countries and millions of them. And then I said to him, when you genetic engineer something, it's not just that single gene and protein that's produced. There's a human gene cell program. It was a cystic fibrosis study. They put a single gene into the DNA, and up to 5% of the functioning genes changed their levels of expression, which means you can have new toxins or new allergens or new carcinogens. Shouldn't you test the entire composition of the saliva in the mosquitoes that you're releasing? And I'll never forget his answer. He said, good idea. So these are the brain cells behind the people who are willing to alter our gene pool irreversibly. And for years, we didn't have any examples in genetic engineering, but we did have the rabbits in Australia. 24 rabbits released in 1859 so that settlers would feel more at home. By the 1920s, there was over 10 billion because rabbits multiply like rabbits. Now imagine taking an ecosystem and with the gene rush going on, replacing many, many organisms in that ecosystem in ways that you cannot predict. And then if they're microbes, they travel around the world, they mutate, they swap genes, and now your little release is changing the nature of nature for all future generations.
Yeah, it is terrifying. And all right, well, tell me what the good news is.
Okay, so our Institute for Responsible Technology is starting a movement to pass laws to prevent the release of genetically engineered microbes. I've been building movements for decades. We built the consumer education movement, and there's a lot of support. And I'd like to invite people to go to responsibletechnology.org/take action and be part of our support team. Right now, we have a letter to Secretary Vilsack. Last month, we had a comment period comment. We're going to have a declaration coming. So it'll be an opportunity to check in with that page over time to lend your support and also to make a donation to our nonprofit. But the idea is that there's many groups out there that already depend on a healthy microbiome for their success. The medical doctors now realize how important the microbiome is. And so that community and their patients are one natural ally. Regenerative agriculture is a natural ally because the microbes in the soil do the heavy lifting. The climate change movement is a natural ally that they want to sequester the carbon in the soil. They need the microbes healthy to do that. If you release genetically engineered microbes, then it alters that. In fact, people living on Earth are a natural ally because algae produces 70% of the Earth's oxygen, and you want to protect that. We have the environmentalists, the oceans people. We have the indigenous and spiritual people that believe that GMO really means God move over. So we have movements of movements that already know that their success depends on the success of the microbiome and, in a bigger picture, on nature. So we are going to, and are in the process of producing materials that can be disseminated through these different movements so we can all work together. And it's interesting, Robert, that when you think about just how terrifying this is, it's really, as an existential threat, an opportunity. I like to think of patients that are given a really bad prognosis, where that prognosis or diagnosis ends up being a blessing for them because it changes their perspective, it changes their behavior. They make new decisions. Here, unlike the bigger issues of existential threats like global warming and nuclear power, a single individual can impact all living beings for all future generations. Someone in a high school, there's a level of potential damage to nature that individuals have that they've never had before. So it demands a restructuring of our relationship with nature so that instead of simply moving along and manipulating it for our benefit, we are forced to protect it. And as a safeguarder of nature, for this purpose, we become a safeguarder of nature for many areas. Now, I believe personally that consciousness as an individual is not so linear and local. We've seen in history how civilizations all of a sudden adopt something new quickly, often in many different parts of the world at the same time. So I'm hoping that the attention and shift that's necessary to protect us and change our relationship with nature will actually deliver the silver lining on this threat, where we actually realize that we must, in order to survive, honor, protect, and uphold the nature of nature. And so that ultimately is the goal of the Institute for Responsible Technology. And we need to pass the local and national laws, create international treaties, raise the money in the war chest to make it happen. And again, I want to invite people to responsibletechnology.org because we're at a critical time right now to see if we can actually make this happen globally.
Jeffrey Smith, how can people get in touch with you?
Responsibletechnology.org is the mothership of our program. We have a Facebook page, we have Instagram, we have Twitter, we have TikTok. I'm now a TikTok phenomenon. I have all these views on TikTok. Who knew? And I would like to invite people to share this interview with others because one of the issues is if people haven't thought about this, they're not alone. I was talking to one of the leading experts at functional medicine and progressive healthcare. And I spoke to him for about an hour about what was happening. And he said, I'm embarrassed that I haven't spent more attention on this, thinking about the implications. He joined our science advisory board. He introduced us to some scientists to work with. Wherever we go, we realize people haven't thought about it much. In fact, I talked to microbiologists One microbiologist said, "Oh yeah, there's no problem genetically engineered microbes." I said, "Let's talk about it." An hour later, he said, "I totally agree with you. No genetically engineered microbe should be released." And it was a microbiologist who'd been working in the field for decades. We had our staff microbiologist call 5 of his friends and say, "What do you think of the genetic engineering of microbes?" And all of them said, "I hadn't thought about it that much." So this is a disadvantage because it's not being thought of, and it's an advantage because we get to reframe the debate fresh, saying, well, we should think about it. It is urgent, and let's actually do something, because now we've arrived at that inevitable time in human civilization where we can redirect the streams of evolution for all time for the price of dinner if you have a CRISPR kit. It's not a time to just wait and see. We must step up and take the responsibility demanded of this new technology.
Jeffrey Smith, the Institute for Responsible Technology, thank you so much, and thanks for a wonderful conversation.
Thank you, Robert.